Well, hello, AP Bio students. Mrs. Shelton here, talking to you about Chapter 10, Meiosis and Sexual Life Cycles. So, after just covering mitosis, this is kind of a good bridge into our study of heredity, which is going to be our next unit. But what we need to make sure that we are addressing here is understanding reproduction and the importance of meiosis and reproduction and yielding genetic diversity. With this, chapter 10 in your book, also resources you're going to want to make sure that you are using is the test prep review series. This is in topic 4. And in topic 4, it's pages 99 through 105. And then also there's lots of practice questions, interactive questions from the old textbook study guide, chapter 13. These would be really good for you guys to use in order to then review the content. So for today, there's really a lot of just terms that we need to make sure that we can understand when we do talk about inheritance and then reproductive systems and cells that are involved in reproduction. So it's very term heavy, but it's really important that we get some of these really critical terms down so that we can apply them to genetics in the future. First of all, what we addressed, so actually if we look here at this particular family, so this family is kind of the first picture that you see. And what we need to make sure that we understand is that we don't clone ourselves. We reproduce sexually, and most animals reproduce sexually. And so with that, we always get genetic diversity. Sexual reproduction leads to genetic diversity because we can only contribute half of our genetic information to our offspring. So we pass on genes. Genes are segments of DNA that code for a protein. We'll talk about genes later on when we do talk about gene expression, but it's the segments that code for proteins. Well, we know that proteins are everything. So these are our characteristics. Now, when we pass on half of our genetic material, we pass them on in gametes. Remember, gametes have the ability to fuse with other gametes to create new genetic combinations. Now, it's actually through the process of meiosis where we as animals, we produce gametes. Well, that's actually where a great deal of genetic diversity comes from, because what half of our genetic material do we actually pass on to our kids? Well, we can actually look here at these 3 kids. Well, they all look different. They all have similarities. They all have differences. And actually, in life, diversity is a good thing. We do not want a whole bunch of clones unless you are an asexual reproducing organism and conditions are ideal. So we do need to be aware of advantages and disadvantages of asexual and sexual reproduction. So when we say asexual reproduction, what's happening there is we're truly making clones of the organism. With that, reproduction is super fast. So if conditions are ideal for the genetic combination that organism has, well, they can very quickly colonize an area, again, if conditions are good. But if the conditions change, and now that genetic combination is not well suited for that environment, well, asexual reproduction doesn't lead to a lot of diversity for natural selection to select upon. With sexual reproduction, the huge advantage there is genetic diversity. We always get genetic diversity. The offspring is never like the original parents when we have sexual reproduction. And we'll talk about the 3 different ways that sexual reproduction leads to this genetic diversity. Because with diversity, that actually allows for organisms now to have variation within the populations and natural selection to act upon it in order to now have adaptations that are much better suited for each given environment. Now, when we do talk about section 2, so fertilization, meiosis, and alternate life cycles. Here are some terms. I told you that we need to make sure that we're going over some different terms. We need to make sure that we have, in part A, I'm giving you 7 terms that we need to go over. And with those 7 terms, some of them are similar. We've seen them before. So we already know and have the definition of what a somatic cell is. Somatic cells are your normal body cells that contain your normal sets of chromosomes. Now, somatic cells are not our sex cells. So with a somatic cell, somatic cells are diploid. What that means is we have 2 of every chromosome. So we would refer to that shorthand notation of diploid cells. We would refer to that as a 2N cell of two of every chromosome. Now, as humans, we have 23 pairs of chromosomes. So the pairs are referred to as homologous chromosomes. We receive one of chromosome 1 from our dad, one of chromosome 1 from our mom. And this picture that we're actually seeing here, this picture is referred to as a karyotype. So this karyotype was actually a literal picture. So that was taken of a cell that was undergoing prophase, prometaphase, when chromosomes have already duplicated. So we're seeing duplicated chromosomes here. There's actually two sister chromatids held together by a centromere. And so this, we can see the drawing here. The centromere is holding the two sister chromatids together, but this would be my maternal chromosome. This would be a paternal chromosome, but those come together as a pair to form homologous chromosomes. In an animal, all of our somatic cells are diploid. Now, when meiosis occurs, though, meiosis is always going to reduce this chromosome number in half. And what's going to happen in meiosis is just one of each of our 23 chromosomes is going to go into a gamete. So those gametes that animals have, those gametes are referred to as haploid cells. And that really is our definition of meiosis, is the division of one diploid nuclei to 4 haploid nuclei is what happens in meiosis across all eukaryotes. Now, when we also look at our chromosomes, so in this karyotype, you're seeing that in humans we have 23 pairs. And so here our homologous chromosomes are numbered 1 through 22. They are not identical. So chromosome 1 is not identical to this chromosome number 1. They carry the same types of genes. However, they carry different variations, or what we'll talk about in genetics, different alleles of that particular gene on there. So if this green band here is coding for genes for eye color, well, maybe this one is coding for brown and brown colored pigments for eye color, and this one is coding for blue color pigments and eye color. So they carry the same types of information, but do not get under the impression that homologous chromosomes are identical. They're homologous. So you also see the banding pattern when it's stained, is they're stained the same way because of the fact that they're carrying very similar genes on each of those sets. Now, when I also look at these 22 chromosomes, they actually are arranged based on what genes and based on size and banding patterns. I have 22 pairs. These are what are referred to as my autosomes. So these first 22 pairs of chromosomes, when we line them up, they are autosomes. These are the same in all males and females. So all males and females are going to have the same combination of homologous chromosomes that are autosomes. However, we have a pair of chromosomes that are referred to as sex chromosomes. So these sex chromosomes in this karyotype, when I line them up, what I'm actually seeing is that this individual has an X sex chromosome and a Y sex chromosome. So in humans, I actually know that this is a male. That's what determines gender. If this was a female, I would see 2 X sex chromosomes in here. But I'd always see, whether it's male or female, I should see the same 22 pair of autosomes. Again, this is my karyotype, is what I'm looking at, showing my homologous chromosomes in a diploid cell. Now, when we talk about meiosis, in meiosis, what has to happen in meiosis is these diploid chromosome pairs have to separate. Now, it all starts at the end of S phase, G2 phase, just like we talked about with meiosis. All the DNA has copied, each duplicated chromosome is holding on to its sister chromatid, but what ends up having to happen in meiosis is we actually have to separate our homologous pairs of chromosomes, and then we have to separate our sister chromatids. So each new cell ends up actually having only a fourth of the genetic information that the original cell had at the beginning of meiosis, of that cell division. Now, in meiosis, so when we talk about meiosis, again, whether we're talking about plants, whether we're talking about fungus, whether we're talking about animals, meiosis is always a reduction of your chromosomes from a diploid nuclei to 4 haploid nuclei. Now, in animals, that process of meiosis is producing gametes of egg and sperm. Some terms that we need to make sure that we are also addressing here. So meiosis is going to be reducing the genetic material into a haploid nuclei. But we also need to make sure that we understand that gametes, so gametes are signified as just N, where a diploid cell is referred to as 2N. So this is showing you that there's 2 of every chromosome. This is showing you there's 1 of every chromosome for that organism. For us, our diploid number is 46. That means we have 46 total chromosomes in each of our somatic cells. In our gametes, our haploid number is 23, meaning that each of our gametes should have 23 chromosomes in it. So every organism, though, that is diploid, it's going to be signified with this signal, this shorthand notation showing 2N, that it's a diploid cell, or a single N shows that it's a haploid cell. Now, not all haploid cells are gametes, though. When it's a gamete, like we talked about last chapter, gametes have the ability to fuse with other gametes to create new combinations of genetic material. That process of gametes fusing together is called fertilization. So that's a term that we need to make sure that we understand. And anytime fertilization occurs, we're going to be combining the genetic material of those gametes in order to now create a new genetic combination that's referred to as a zygote. So make sure those are terms that we understand. We know that meiosis is reducing our diploid number to haploid numbers in our cells. Gametes then, when they fuse together, occur in the process of fertilization in order to now create a new genetic combination that we refer to as a zygote. Now, a skill that you're going to have to make sure that you understand is understand different life cycles. So with different sexual life cycles of an animal versus a plant versus then a fungus, there's actually different times when the multicellular organism is diploid or haploid. So just for example, for animals, all of our somatic cells, our multicellular state, all of our cells are diploid. There's 2 of every chromosome. But actually in a fungus, all the somatic cells are actually haploid. So if we went and picked off a cell from a mushroom and looked at the chromosomes and analyzed the chromosomes when they're undergoing prometaphase, metaphase, well, then we would actually see that all of them have 1 of every chromosome. So that's a huge difference between an animal versus a fungus of what those somatic cells, the fact that they are all haploid. Now in plants, the multicellular state, and so that's what we're looking at is the multicellular state. The multicellular state of a plant is both diploid and haploid. Plants undergo what is referred to as an alternation of generation, where one generation is diploid, they reduce their chromosome numbers through meiosis and create these haploid cells that are referred to as spores. Now, a spore is different from a gamete. And you're actually going to see that there are spores that are also produced then in fungus. A spore is a haploid cell that can grow into a multicellular organism. Now, a gamete, though, is a haploid cell that can fuse with another gamete to create a new diploid zygote. So there's a big difference in what those are. So in an animal, if we actually just take a look at our normal sexual life cycle, we understand this very well, where we are a diploid multicellular organism. Meiosis produces gametes for us that are haploid, and then fertilization, 2 haploid gametes fuse together to create our 2N zygote that then develops into the trillion cell multicellular organism of animals. That's our life cycle. Now, plants. With plants, this is referred to as alternation of generation. And for alternation of generation with plants, let's start with the diploid multicellular organism. This diploid plant, it's going to undergo meiosis, but in meiosis, these haploid cells are referred to as spores. So this is why this plant is referred to as a sporophyte. So sporophytes produce spores, and a spore is a haploid cell that can grow into a multicellular organism. So sporophytes undergo meiosis to produce spores. That spore now can grow into a multicellular plant that is all haploid. But now for this plant to reproduce, the only way this plant is going to reproduce is to produce gametes via mitosis. Because remember, mitosis is making identical nuclei. So here, if all of your cells are already just one chromosome each, well, your gametes need to be identical copies, but they can only now reproduce by fusing with gametes from a different gametophyte plant. So my gametophyte is a haploid multicellular organism that produces gametes, but those gametes are produced via mitosis. So then when fertilization happens, two gametes fuse together to produce another diploid zygote. Well, now I have a new genetic combination. That diploid zygote grows into a sporophyte that's diploid, and then for it to reproduce, it undergoes meiosis to produce then spores. These spores grow into a multicellular organism that then undergoes mitosis to make gametes. So it's a gametophyte, makes gametes, and then fertilization. So this is alternation of generation. And actually with plants, they kind of end up having the best of both worlds. As far as with meiosis, anytime meiosis happens, we are getting genetic diversity. We'll talk about that why through the process, but then they also have the ability then to colonize and reproduce very rapidly as well. Now, in a fungus, what's different here about a fungus is that they don't have alternation of generation. We're never going to see a multicellular state of a fungus that is actually diploid. So what actually happens, we'll start with the multicellular state of a fungus. It's haploid. So since it's haploid, with this particular fungus, well, when it makes a gamete, it still has to make gametes in order to reproduce. But what ends up happening is it, again, like plants where we had my gametophyte, it undergoes mitosis to produce gametes. But for a new fungus to develop, this gamete has to fuse with another gamete from a different fungus in fertilization to produce a new genetic combination of zygote. But what ends up happening with this zygote is this zygote will almost immediately undergo meiosis in order to produce spores. So these spores then, these haploid spores, then can move to lots of different places and fly by the wind. And those spores then can grow into the multicellular fungus that then, once it's multicellular, reaches its reproductive age, it produces gametes through mitosis that have to fuse with other gametes to create a new offspring. So those are my different sexual life cycles, and I need to make sure I'm understanding the difference between an animal and their sexual life cycle, plant and their sexual life cycle, and then fungus. The key is always to look at what's the multicellular state. So if we saw this unlabeled and you saw that the only multicellular state that existed was in the haploid state, you would know that it's a fungus. If you saw that the only multicellular state existed in a diploid state, we would know that this is an animal. If you saw the multicellular state of that organism is sometimes diploid and sometimes haploid, then you're going to know that it's a plant. So make sure, but in all of them, because they all sexually reproduce, there's always meiosis, there's always fertilization. And this is actually where we are getting our genetic diversity. Anytime meiosis happens, we're getting genetic diversity in these gametes that are produced. And anytime fertilization, and we actually refer to it as random fertilization, because it is, which gametes come together, it's random, we get more genetic diversity in these cases. So meiosis always produces genetic diversity through crossing over and independent assortment. We're going to talk about those in a little bit, and through fertilization, through random fertilization. Now, when we do address and talk about meiosis, meiosis happens in 2 rounds. So we're going to have prophase, metaphase, anaphase, telophase, cytokinesis. And then we're going to have a second round right afterwards is what happens during meiosis. We're going to have prophase, metaphase, anaphase, telophase, cytokinesis again in order to go from a diploid cell, so a diploid cell with duplicated chromosomes, to 4 haploid cells. The major event, so we actually say that because it goes through 2 rounds, we refer to it as meiosis 1 and meiosis 2. Now, here's the major difference between the first round of meiosis versus the second round of meiosis. And make sure you write this down. In my first round of meiosis, the goal is to separate homologous pairs. So it's those pairs of chromosomes. When I've got pairs that have their sister chromatids attached, what happens on the metaphase plate is that homologous chromosomes line up 2 by 2. And my first round of meiosis is about separating homologous chromosomes. My second round of meiosis, though, is now about separating the sister chromatids. So those are the major events to keep in mind. That's the big picture, and keep those going. So here's actually just meiosis 1, meiosis 2, and what we're seeing here is that sister chromatids are staying together in metaphase. So in metaphase, my homologous pairs line up, and then my homologous pairs separate, and then I immediately have another round of meiosis, and now my chromosomes are going to line up single file, my sister chromatids are going to separate. So let's take a look at some of the things that actually happen here. So meiosis 1, what's going to be happening in meiosis 1, in prophase, homologous pairs are actually going to join together. Well, what happens in this particular case, and this is a really, really important event that actually occurs during prophase. Well, I've got 4 chromatids here that actually all kind of jumble together. So this forms what's referred to as a tetrad. And so that tetrad of 4 chromatids all over arranged, well, they overlap at times, forming this chiasmata. Well, where this overlapping happens, it's random every time. And what can happen is pieces of homologous chromosomes can actually switch places. So they can be from a maternal chromosome and a paternal chromosome rearranging to give new combinations of sister chromatids. So this happens in prophase 1. This is called crossing over. This is a huge event that leads to genetic diversity. Now, the second thing that actually happens is my homologous pairs line up 2 by 2. Well, when they line up 2 by 2, well, that's actually going to determine which direction the homologous pairs go to for the new cells. This is called independent assortment. The way this homologous pair lined up has no bearing on how the next homologous pair lines up, but does play a huge role in the delivery of the genetic combinations to the new cells. So right here in prophase 1 and metaphase 1, this is where a tremendous amount of genetic diversity is established. Now we can see that in anaphase 1, my homologous pairs, you're even seeing kind of these little hybrid, I've got blue and red chromatids here from crossing over, and I also have more red going here, more blue going this direction. That is my independent assortment that's leading to that difference. It's random every single time. Crossing over is random, independent assortment is random, and so we get new genetic combinations in our gametes every single time. This is what you need to be looking for in order to determine in a picture if we're in meiosis 1, or if we are in meiosis 2, or if we're in mitosis. If you see chromosomes lining up 2 by 2, or if you see homologous chromosomes separating, we know that we are in meiosis 1. So here we're seeing it a little more closely. Now we're seeing that separation of my homologous chromosomes in meiosis 1. But then what happens now is meiosis 2. And in meiosis 2, I'm not getting any genetic diversity happening in meiosis 2. What's happening in meiosis 2 is now I'm separating my sister chromatids. So I can end up creating 4 haploid cells. So this is going to look very similar to what we learned in metaphase of mitosis, where your chromosomes just line up along the metaphase plate. Sister chromatids move to opposite poles because of the motoring action of the kinetochores. And then I get 2 new nuclei forming from each of those, but really now I'm getting 4. Here's a closer look at crossing over. And again, crossing over happens in prophase 1 of meiosis when my homologous pairs are lining up and pieces of homologous pairs are switching places. So this again is a huge source. Where they switch places, where they interchange, it's random every single time. So here's a compare and contrast of mitosis versus meiosis, and making sure that we are aware of the major differences that happen. Remember that in meiosis, I'm going to have 2 rounds of cell division happening. I'm going to start with diploid cells that have already replicated their chromosomes. So each chromosome is going to have its sister chromatid attached to the centromere. But remember in metaphase 1, I'm going to have my pairs of homologous chromosomes lining up, So they're going to line up 2 by 2. And it's through this process of prophase and metaphase where I'm going to have crossing over. And then how they line up in metaphase, this is my independent assortment. And in my metaphase 1, they line up 2 by 2. Which direction they line up doesn't matter. And then my homologous pairs separate. And then in my second round of meiosis, now I'm separating my sister chromatids. I only have 1 round of cell division in mitosis in order to produce 2 identical nuclei. My nuclei that are produced in meiosis should not be identical. They're all identical in chromosome number, but they are different in which genes they have. So this is just kind of that summary as we look through, making sure that we're understanding just our differences between mitosis versus meiosis and being able to recognize where independent assortment and crossing over leads to genetic diversity. So my crossing over, pieces of homologous chromosomes switching places. So this is crossing over. This is a huge source of genetic diversity. But then with independent assortment, how my chromosome pairs line up during metaphase 1, this is going to determine different combinations of maternal and paternal chromosomes for the offspring. And then finally, when we actually add to it, when we add now that these gametes are going to end up now having to join with other gametes that were produced via crossing over, independent assortment with their genetic diversity. Now random fertilization is going to create a tremendous amount of genetic diversity in the offspring. And again, the huge advantage of genetic diversity is more genetic combinations that can now survive changing environments. And this is a huge part of then understanding the natural selection process. So with that being said, that is your overview of chapter 10. You guys just heard the bell ring. So I'm having students coming in right now this is the B group of students coming in to take their chapter 9 test. But this is chapter 10 on meiosis and sexual life cycles. So I will see you guys in class to go over more of this, to answer questions, but hopefully you have it. There's your announcement there. All right, see you guys later. Bye.